This is Mrs. Palmer-Quay, and this video covers the few learning targets that were specific for honor students but recommended for everyone else. The first one covers this idea of neoplasm, and I really should have mentioned this in the previous video. It's just the fancy scientific name for tumor, and the word neoplasm means new tissue, for those of you that know your prefixes. So a neoplasm is an abnormal mass of cells arising from one uncontrolled mitosis. One cell just continues to divide when it should be differentiating, so that the cells are clones of the original in a neoplasm. It may be benign or it may be malignant. So some of the characteristics of a benign neoplasm is that the cells are relatively differentiated. They're pretty close to the cells that they came from originally, to the tissue that's surrounding them. Benign neoplasms also tend to be slow growing, not always, but usually, and they are localized. So examples are uterine fibroids, various moles, lymphomas, which are fatty tumors. Some benign tumors tend to become malignant over time. Colon polyps are a perfect example of that, which is why people are recommending that you have a colonoscopy after you reach age 50, and if there are any polyps, they go ahead and remove them. So just some examples here. We have sort of your standard mole. This is a mole as well. The dark cells are hidden under the surface layer, and it just bumps out. And then you see here the fatty tumor on the back of this person. The lymphomas are very common among dogs. You might know of them, be more familiar with them with dogs, but they occur in people as well. A malignant neoplasm has very undifferentiated cells. They don't look anything like the original tissue where the cell originated from, or the tissue that is around it. They are graded on a level of 1 to 4, 1 being not being fairly differentiated still. 1 is the best. 4 would be ones that are very much undifferentiated and are just busy dividing. Malignant neoplasms usually are growing rapidly, and the cells are prone to break off and travel through the lymph vessels to start new growth in other areas, calling metastasizing. So here's some example of skin cancers. This is basal cell carcinoma and then melanoma, two terms that you're probably very familiar with because skin cancer is a concern today. The second learning target is on amitotic tissues. Again, if you know your prefixes, A here means not, so they're not mitotic anymore. They were mentioned briefly in the previous videos. These are tissues that do not undergo mitosis after the growth period of the organism has ended. And in human, you find these types of tissues in the brain, the heart, muscle, and the retina on the back of the eye. The primary reason that these tissues do not undergo mitosis at maturation is because mitosis would compromise their function. Just think about it. In your brain, you have many, many synapses between all the neurons, and part of the job of those synapses is to give you memory. We're not quite sure how this all works, but if you had cells that would divide, and along with cell division, then you also have to have cell death, you would be losing your memory slowly over time. Or think about the heart. The heart starts beating very early on in development, and it doesn't stop until your life is over. Well, heart cells can't take time out to do mitosis. When a cell is undergoing mitosis, all of its normal functioning stops during that time period of dividing, and so that just wouldn't work for your heart. Muscle cells, as you will see as we get into the muscular system later on this year, are very interesting. The skeletal muscle cells are multinucleated, and that's because a large number of cells have merged together. So again, it's not a kind of cell that could divide easily. And then again, the back retina of the eye is a place that is rich in nervous connections, neurological connections, because of all of the information coming from the rod cells and the cone cells. So again, if you had mitosis going on there, you would compromise the function of this organ. Unfortunately, tissues that are amitotic can have severe repercussions if they are injured severely, because they're not able to reproduce more cells of the same kind, so that all you are left with is scar tissue. And then the last learning target I want to briefly discuss is tissue repair. There was another one on tissue function and location, but that was pretty much covered in the previous video on tissues. So tissue repair will lead to two things. If you have mitotic tissue that will regenerate, then you will have the replacement of the part that was injured and removed. It'll be regenerated and replaced by cells. If the tissue or the tissue is amitotic or the injury is severe, then you are going to have, instead of regeneration, you'll have scarring, which the fancy word for that is fibrosis. So the end results depends upon the site and the severity of the injury, what kind of tissue was involved with it, and how badly was the injury. The first step in tissue repair is hemostasis, and hemo refers to the blood, stasis is referring to being stable and unmoving. So the first thing that we want to have happen in a wound repair is to stabilize the bleeding or stabilize the blood situation. And most of the wounds that we do experience on an ordinary day-to-day basis are in the skin, in the epidermis, and this is a mitotic tissue, so repair happens fairly regularly. So hemostasis, stabilize the blood. The first thing is that these little blood cells, they're not really cells, bits of cytoplasm and membrane called platelets come into action, and they look like little plates in the unactivated state, but when the broken fibers of connective tissue are formed as the wound is opened up, as the tissue is breaking, then various chemical signals go out and the platelets change shape and become these sort of that little feet sticking out. They become more sticky and they can hook together to form a clot and seal off the damaged blood vessels. So the very first thing is that we stop the bleeding, the platelets come in and plug the hole, and then the rest of the steps can happen for wound repair. So after you have the bleeding stopped, the mast cells, which we've heard about before with connective tissue, release this histamine, which causes capillaries to become leaky. Plasma leaks out into the tissues along with a number of proteins and various other important chemicals and triggering factors. And they also bring out the neutrophils, which are one of the white blood cells that are involved in cleaning up any bacteria, to cleaning up the dead tissue that had been destroyed by whatever situation caused the wound. In a few days, you have macrophages also coming in and to phagocyze any bacteria, again, any dead tissue. So the cleanup crew comes in during the inflammation stage. So after the inflammation stage is over, we move into proliferation. This proliferation means multiplying, so this is when you sit to see some new things being added. The blood clot is replaced by granulation tissue, which you will probably recognize as the pebbled red tissue that's there underneath your scab if you pick your scabs open. Fibroblasts, the connective tissue cells, create a bridge of collagen fibers to help stabilize the wound, and they eventually will contract and pull the wound together. The macrophages again go in, this time to digest the clot to allow for cell regrowth in the space that's underneath. If the wound is in mitotic tissue or the injury or infection is mild, then you see new cells begin forming below the scab fairly easily. And when you're done with complete healing, there may be a tiny line of scar tissue, but there's really not much scarring. But if the tissue is amitotic or the injury or infection is severe, then the granulation tissue really becomes a fibrous tissue patch instead of a new regenerated tissue, and eventually it becomes all collagen. So that heavy scarring is the result. The tissue is quite strong, but it can't perform the original function. In the case of amitotic tissues, it really might get in the way. In other cases, you can have the scarring causing adhesions, which can lead to problems. Abdominal surgery often causes adhesions between the organs in the abdomen and then may cause obstructions in the intestine. Joint surgery sometimes might cause adhesions that would even immobilize the joint. So heavy scarring can be a problem. And finally, you have a time period of remodeling and maturation, where the growth of the connective tissue continues below the surface to strengthen and support the new cells. This can take up to 2 years to complete, so it can be quite a while after a wound, especially a significant wound, before the tissue is all strong and back together again. So here's a little chart that goes through the steps of wound healing, starting with the injury. You see we've got some skin missing on this surface wound, and the platelets are coming into the rescue to get ready to provide the first step, the blood clot. So we have a blood clot forming between a combination of platelets and fibrin. keeping the, stopping the bleeding and allowing some time for these various immune system components to be part of the early inflammation response. So these PMNs are just another way of saying the neutrophils, which I mentioned, a type of white blood cell that will come in to clean up dead tissue and eat bacteria, followed by the macrophages doing the same type of job just a little bit later in the time period. You can see this particular chart sets things as early inflammation and then late inflammation. And then after about 3 days we start seeing proliferation. The fibroblasts are activated. They're building more collagen, connecting the sides of the wounds together, providing a scaffolding to put new cells on, new epidermal cells. And then eventually, weeks or months afterwards is the time of remodeling, where the new cells have filled in the space, but still we have some development going on underneath among the strengthening of the collagen fibers and the other connective tissue. So that covers those few learning targets that are required for honor students, but they're important topics that would be at least, I think, of interest, even if you don't memorize all the information, for everyone in the class.